Hi, hello everyone. I'm Raj representing from Group 5. This is our Chapter 1, Week 1, Introduction to Biomol, that is Molecular Biology in Conservation. So, the objective is to conserve energy, species, and habitat on the Earth and find the molecular biology technology in our biomolecule. And for first, the second week is goes for the introduction and DNA and gel electrophoresis. So the introduction part we will learn about central dogma and degeneracy and electrophoresis. That means we have to identify what is gel electrophoresis and agrogel. And we will go for the week 3, that is the next chapter, which is polymerase chain reaction, also known as PCR, and dominant markers. So we will identify the molecular markers and biomarkers, and also the PCR chain reaction, what is needed for the PCR cycle, that is PCR cycle, template, primer, enzyme, magnesium 2 plus, and so on. And we will go on for the week 4, that is RAPD. RAPD is actually random amplified polymorphic DNA, which is called RAPD markers, RAPD band crossing, cluster analysis, consistency tree. And we will go for week 5, which is a microsatellite, which is using a microsatellite, also known as SSR, which is simple sequence repeat, and STR, short tandem random, that is genotypic codominant markers. We will identify what using a microsatellite, how we will finding the bending pattern, segregating profile, DNA analysis, and we'll go for mitochondrial DNA, which is a mitochondrial DNA. What's the evolution and uses? So we will identify what's the usage of mitochondria DNA, the shape, location, the function, the microdn DNA versus nuclear DNA, and the conservation, what we use in conservation for plants, seed, animal, microbes, and what is the definition of a molecular markers, and what's the function of a genetic diversity. And we'll go on for the genetic, also known as ribosomal DNA, and the evolution is, which is a ribosomal DNA, what's the function, shape, location, when the function of a ribosomal DNA, and mutation, what type of mutation it can be found in the ribosomal DNA, all about it, and SNP, and followed by the molecular markers, got about two types, that is RFLP and okay. For week 8 we have a bacterial DID, which is using a identification using 16S rDNA, and RAPD AFLP, which is using what is function of 16S rDNA, what is RAPD, and you have to know what is a AFLP. So we also known as what we need for RAPD primers, advantages and disadvantages of RAPD, and also advantages and disadvantages of AFLP. For week 9, there will be DNA barcoding, processing, and reprocessing. So the processing of DNA sequences, also we have to find out the output data from DNA sequencer, which is primary data, secondary data, or tertiary data, followed by keeping DNA sequences using from gene bank or BOLD, also known as barcoding of live database, and followed by basic of phylogenetic analysis, making inference from DNA data using from philosophical data, either from the cladistic or phylogenetic tree, and the next second one will be mechanical. Then the third one will be DNA data matrix using sequence alignment, either be identified homology or mismatch point of mutation, whether there's about four type, which is also known as for substitution, insertion, deletion, and inversion. The last will be terminology in phylogenetic analysis, either using a sequence alignment or phylogenetic tree. So phylogenetic tree, there's a lot of branch you can find it, which is root, branch, node, tip, clade, common ancestor, bifurcating, lineage, branch length, distance between. So the week 10 will be molecular phylogenetic analysis, which is one, where there will be distant metric method. First is NJ, the neighboring joint, and UPGMA, first calculate distance in the internal nodes, and for UPGMA is construct cluster. The following will be character based method, which is the for three type: maximum parsimony, second is maximum likelihood, third one will be Bayesian inference BI, then the third one will be validation method using bootstrapping pseudo repeat. There's software mega, yeah, okay, and followed by week 11, yeah. Okay, we will go for week 11, that is molecular phylogenetic analysis 2. That is, we will look on for the maximum likelihood approach, which is a statistical approach, and the components will be using a data model or criteria to move from the data and the model to an estimate of the parameter of the model. And we will go for week 12. There is application of molecular phylogenetic analysis in conservation 1, part 1, where we will be checking with the phylogenetic relationship for taxonomy, where we using a DNA sequencing or DNA barcoding, followed by phylogenetic diversity. We will use a PD, where we will have that those you can see the equations, where we use PD to find the rarity aspect, richness aspect, phylogenetic diversity as a proximate for functional diversity or evolutionary potential. Yeah, as you can see there, yeah. And we will go on with the week 13, where application of molecular phylogenetic analysis part 2, where we will have a three part. First part is dating phylogenetic 3, where we will use molecular clock concept or putting time on the tree, and the second concept we will see phylogeography, where we will see geographically structured genetic signal within and this among the species. A third concept will use a phylogenetic signal, where there's a tendency for relating species to resemble each other. And for the last week, we will check it out with the case study 3 of the use of molecular technique, which you guys or we will be see soon, which is coming soon. Yeah. Okay, so altogether we have total six tasks. There's actually eight tasks, but so far we have stated about seven tasks. The first task is actually we have to take read a journal and read about it. And task two, where we will have to find the taxonomy method and materials from that journal, and followed by task 3, where we will have to define the PCR method, the gene, and the software used for alignment from that journal where we are study. And the task 4, where we will given a gel for electrophoresis, where there are four or five things, then we have to compare and contrast from the gel electrophoresis. For task 5, we have to obtain a gene bank account number from our journal, where we have to find the gene from that gene bank, and from there we have to have a sequence alignment. We have to do a sequence alignment using bioedit software. And then followed by task 6, where we have to make a phylogenetic tree using mega software from the journal we study. And task 7 coming soon. Yeah, made it out. Hi guys, okay, then for tutorial, we have a tutorial 1, where we will study about PCR, where the PCR, what the function, everything, all about PCR for the tutorial. Second tutorial, we will study about gel electrophoresis, what main components, what all the details from the gel electrophoresis. Tutorial 3, we will identify of bacteria using 16 rDNA from gene sequencer, where we have to use gene bank to get the genes all from the gene bank. And for tutorial 4, we will construct phylogenetic tree using bioedit. Tutorial 5, where we will see phylogenetic analysis, where MP/ML we will using from the Mega software to get the tree, all about that. So we will learn from the Mega software. And tutorial 6, which is going on now, you can see my sounds all going on, where we have to reconstruct our biomol structure, where we have to check all did it, and task 1 until task 6, tutorial 1 until 6, and assignment which is and which are going on now, where you can listen to here. So once again I say thank you for all to listen all about us. This is our project for task 7 6 task 7 and 8. We will record all about it, and hope so we will get a good mark. And once again, behalf of our member group 5, I am saying thank you, sir. Hi, this is it, and hey, yeah, thank you.